Greetings everyone, my name is Thabile Madlala and I'll be doing a presentation on vaccine design against Toxoplasma gondii using surface antigen 1 and dense granule protein 6 antigens through immunoinformatics approach. Toxoplasmosis is a zoonotic disease caused by Toxoplasma gondii, which is an obligate intracellular parasite resulting in congenital infections and abortions in humans and livestock. This parasite is normally found in cats, which serve as a primary host or carrier of the parasite, and other animals, which often serves as intermediate hosts. The parasite threatens welfare of animals and public health due to increased zoonotic risk of transmission to humans, as it is transmitted via consumption of contaminated food and water, or being in contact with contaminated dust. In humans, this parasite can be transmitted by ingesting poorly cooked meat containing parasite tissue cyst or being in contact with contaminated water or vegetables. Hence, the aim of the study was to explore the T. gondii antigens of sheep by identifying antigenic T cell and B cell epitopes and to design a potentially cost-effective peptide-based vaccine through immunoinformatic techniques. Now, this was achieved through a series of computational steps, including a retrieval of amino acid sequence of sheep Toxoplasma gondii surface antigen 1 and dense granule protein 6 from NCBI. These sequences are subjected to multiple sequence alignment using cluster W and transmembrane structural analysis to determine or generate conserved sequences. The conserved sequences are further used as input file in an online server called Immune Epitope Database and Analysis Resource, known as IEDB, to generate our T cell, which are CD8 and CD4, and B cell epitopes. Now, to confirm whether the predicted T cell epitopes were antigenic and 100% conserved, immunogenic, or could not cause any allergic reaction to oats, the predicted epitopes were assessed for antigenicity, immunogenicity, conservancy, and allergenicity. CD4 epitopes were further assessed for interferon gamma and interleukin-inducing properties. After these analyses, the epitopes found to exhibit these properties were regarded as our final predicted epitopes, which were then used to design our multi-epitope vaccine construct, which further underwent a series of validation analyses. Okay. We were able to identify 2 T cell epitopes and 10 B cell epitopes, which were joined together using GPGP and KK linkers to form a multi-epitope vaccine construct as seen in Figure 1. A cholera toxin B antigen was also attached to our construct in order to enhance its immunogenicity. Now, the texture and structure of our vaccine as seen in Figure 1 was predicted using an online server called 3D Pro and further refined using a Galaxy Web Server. The quality of our design model was assessed using PROSA server, where a Z-score of -2.93 was observed, indicating a good overall model quality of our vaccine. The design of vaccine was further docked through CHPC platforms with a toll-like receptor, which is TLR4, to assess the vaccine stability and interactions with a receptor. Where of the 50 binding conformations produced, a model selected had the lowest binding affinity of -151.654 kcal per mole, as seen in Figure 2, suggesting proper binding and interactions with TL4, and further confirming the stability of our vaccine. The vaccine structure was further assessed for physiochemical properties, which revealed our vaccine to have a molecular weight of 34.08 kilodaltons, to be antigenic with a score of 0.9135, soluble with a score of 0.9485, and also observed to have a theoretical PI of 9.8 and an instability index of 29.36. These properties classified our vaccine to be antigenic, basic, and stable. The aliphatic index and grand average of hydropathicity, which is the query, were observed to be 75.62 and -0.425 respectively, showing or further confirming our vaccines to be thermostable and hydrophilic in nature. Now, the current study designed a promising multi-epitope vaccine consisting of a diverse T-cell and B-cell epitopes from T gondii antigens. The design vaccine was highly antigenic and non-antigenic, showing great promise and qualifying properties for production. Moving forward with our study, we still have to compare the structural stability of the proposed vaccine against toll-like receptor, TLR4 and TLR2, through molecular dynamic simulations to be performed on the CHPC platform, and to assess the ability of the vaccines to trigger immune response through immune simulations. These findings will be very valuable to the future studies focusing on vaccine development against Toxoplasma gondii. Thank you.